Our next speaker is Dr. Kellie Smith from Johns Hopkins University in Baltimore, Maryland. I'm actually super excited to introduce Kellie, as this is a bit of a virtual reunion. We were both in Dr. Ronaldo's lab at Pitt, overlapping for a few years. Kellie entered the lab through Pitt School of Medicine, and her focus was on T-cell immunology and immunotherapy. During her fellowship training at Johns Hopkins, she worked under the mentorship of Dr. Drew Pardahl, identifying the correlates of response to anti-PD-1 immunotherapy. She joined the faculty in early 2016 and has since collaborated with many clinicians within Johns Hopkins and at outside institutions on immunotherapy clinical trials aimed at improving treatment options, preventing disease reoccurrence, and understanding the predictors of response to treatment in both early and advanced stage disease. Dr. Smith's group has also developed a novel patent-pending technique to detect and monitor anti-tumor T cells using a simple liquid biopsy approach called MANAFEST. So I look forward to hearing more about these as she discusses the tumor reactive T cell functional programming in human cancer. Kellie?
Thank you so much, Emily, and thank you guys for having me. I'm really excited to share all of the exciting work that we've been doing. So I thought it would be good, just because I know we have some folks on here with different backgrounds and different areas of expertise, to kind of introduce myself and the main kind of theses of my laboratory. We really have three primary research goals. The first, which is more something for academic interest, is we really want to understand how checkpoint blockade impacts the immune system. And we use a variety of multi-omics technologies to look at this. We are also interested in, not only characterizing these global or systemic effects, but also really understanding how checkpoint blockade impacts the functional programming of antigen-specific T cells. So not just looking at T cells that are in the tumor or T cells that expand in the periphery, but really let's look at the T cells that are arguably the most important to the anti-tumor immune response, and that's T cells that recognize the tumor. And then, of course, of clinical interest, we really want to identify things using these approaches that can associate with clinical outcome and hopefully impact patient care. So I'm just going to jump right in. We all understand the importance of neoantigens and the association of tumor mutational burden with response to IO. And Dr. Mansfield, I think, did a really great job summarizing these in the previous presentation. A few things that I do want to point out is that while there are associations between, number of predicted neoantigens and response, there really have only been a handful of studies that have really fully characterized neoantigen specific T cells. And usually what it winds up being is, you have this very elegant clinical trial or this very elegant scientific study, and at the end, you might have, one or two patients where they or we identify neoantigen specific responses, and all of our publications do that as well. So given that there is this perceived association of neoantigen specific T cell responses with clinical outcome to checkpoint blockade, we really wanted to undertake a massive study looking at global responses in these patients. So at a global level, not just in one patient, one responder, one neoantigen. We really want to get the full picture. So, these foundational studies on the left, the RISV paper, and the right are Neoadjuvant study in lung cancer that had this correlation between percent residual tumor and the number of predicted neoantigens. We really wanted to follow up on these to really understand what's happening to these T cells in these patients. And I always like to add in this little roulette example because I love it and I think it's really clever, and it really underpins our philosophy when it comes to neoantigens. So you have all of these immune responsive neoantigens in patients with high tumor mutational burden, right? So the likelihood of a T cell response being generated is higher. But in patients who generally don't respond, you might only have, one or two immunotherapy sensitive neoantigens. But we think that it really is the quality of the neoantigens, not necessarily the quantity. All it really takes is one good neoantigen to have a successful immune response. And if we can identify patients who have these quality neoantigens, and if we can really functionally profile the T cells that are recognizing them, I think that gives us an immense amount of predictive power. So how do we go about doing this? So ELISpot, you know, I don't want to poo-poo ELISpot because it has served me well over the years. I think during my graduate work, I did like 300 ELISpots or something. So, you know, I love ELISpots. They are super useful. But in general, you know, especially with doing tumor immunology work, there are a few drawbacks. So one of the things is that you can't track the response without repeating the assay. So if you want to do like a pre versus post, you have to do the assay 2 times. When we're dealing with archival tissue specimens, you know, you aren't able to do an ELISpot on archival FFPE. So you need live cells. There are occasions, especially when dealing with patients treated with a checkpoint blockade or patients with IRAEs, you will have some level of background after culture. These assays are cytokine dependent, as we all know, and you aren't really able to look at the clonal dynamics of the response. So you don't know if it's 1 clone or 50 clones that are making up your you know, 100 spot forming cells or whatever it is. So how do we address this? Our solution to this problem, at least with the patients that we look at, was to use T cell receptor sequencing. And why do we want to do that? By sequencing the TCR-β CDR3 region, we are able to track clones that recognize the same antigen. You know, bulk TCR sequencing, a lot of people are using it. It's very ubiquitous at this point in time. And I think some of the data I'm going to show over the next few slides are very interesting. But, you know, the drawback is TCR-seq by itself is not going to inform on antigen specificity, right? So you could just be tracking clones that expand in the periphery, but they're recognizing some virus or you know, maybe the patient got a flu vaccine or something. So that is the caveat with TCR-seq technologies. But the benefit is that you can do this on a variety of sample types. The cells don't need to be live. And basically, if you can get DNA, you can do TCR-seq on the sample. And that effectively creates this DNA molecular barcode that you can track across a variety of tissue types and biospecimens. And this has been associated with clinical outcome, you know, in a variety of studies. And I'm sure you guys are all familiar with these, but a lot of people are now doing bulk TCR sequencing as part of their routine immune studies and clinical trial studies as well. So in order to look into this, we wanted to piggyback on our neoadjuvant anti-PD-1 study, which was published in New England in 2018. So this was a phase 2 study of Nivo in patients with resectable non-small cell lung cancer. They were given 2 doses of Nivo prior to surgical resection. And there was a pretty astonishing 45% major pathologic response rate, and that's identified as having less than 10% residual tumor at the time of surgery. And the study was set up, you know, by immunologists for immunologists. So we were able to get biospecimens at all of these time points, before and after treatment, long-term follow-ups. You know, it really was the dream, not only clinically, but also scientifically. So we're able to look at a lot of these things in these patients. And one of the first things we did was we did bulk TCR-seq on basically every single specimen that we obtained from these 20 patients enrolled to this trial. And some of these findings were published earlier this year. So all of these analyses were done by Jiajia Zhang. She's a data analyst in the Bloomberg Kimmel Institute, where I work. And one of the first things we noticed was this very nice correlation between the productive clonality in the tumor and the number of somatic sequence alterations. So basically tumor mutational burden and clonality, there was this positive association. And when we looked at clonality as a function of residual tumor, so this is the primary clinical endpoint of the study, there was this negative association. So as more tumor was present at the time of surgery, meaning less of a response, the clonality of the T cells was lower in the tumor. And then again, when we want to separate out, on a discrete level, major pathologic responders versus non-responders, if we look in the, if we look in the resected normal lung, you actually don't see any difference in clonality, but really where it is in the tumor bed. You start to see this separation of clonality between responders and non-responders. Then when we specifically look at the highest frequency clonotypes in the tumor, what was also kind of interesting to us, and this is something that we hypothesized but never really had strong enough data to support, is that really the clones that are shared with the periphery are the ones that make this, this difference in clonal space kind of stand out. So if you only look at clones that are found in the tumor, you really don't see a difference in the amount of clonal space that is taken up by these top frequency clones. But if you then look at clones that are shared with the periphery, you again start to see this separation. So what we think is that not only is tumor infiltration important, but it also requires this systemic component as well. So you need T cells that can not only infiltrate the tumor, but can also travel throughout the body. And we suspect this is a mechanism of seeking out micrometastases. Fortunately for the patients, kind of unfortunately, from a scientific standpoint, we don't think we're ever going to reach median relapse-free survival in these patients. We are over 2.5 years out, I think close to 3 years for the last enrolled patient, and only a handful have relapsed. Which is excellent. But, given that, we aren't really able to make any associations of these findings with relapse. So the next thing we wanted to do was use, these newer technologies to figure out the functional programming of these T cells when they're in the tumor and other tissues. So using this TCR as a barcode, we're then able to do single cell TCR-seq, RNA-seq of tumor and normal tissues and identify the transcriptional program of these different clonotypes. So what I'm showing here are UMAP plots of neoadjuvant treated patients. The left is tumor from responders, and the right is tumor from non-responders. And what you can see almost immediately is the red dots, which are representing clonotypes that have expanded from the first dose to the second dose. From second dose to time of surgery, is the clones that are expanding actually reside in different clusters between responders and non-responders. And anyone who knows single cell data knows that we could talk for probably 5 hours about these data, so I'm just going to give a brief overview right now. But when we look at gene expression, what it seems to be is the clonotypes that are expanding in our responders have more of an effector phenotype. So they have granzyme K, they have upregulation of MHC class II and eomesodermin. So there are these functional differences that we're now in the process of teasing out a little bit more to understand, what is different about these clonotypes that are expanding in the periphery between our two response categories. In one of these patients, this is a patient that we've characterized extensively, and I'll show a little bit more data from him in the next couple of slides. But this patient had 4 clones that expanded in the periphery and were found in the tumor. 3 of them had this cytotoxic phenotype. So if we look at genes that were upregulated in these clones, they more or less have to do with cytotoxicity. But then the 4th clone was incidentally a Th17 clone. So, again, these are the highest frequency clones in the tumor. They're expanding in the periphery. But what if they have these opposing, functional attributes? How does that impact the biology and the clinical outcomes? So these are all things that we're kind of exploring in this setting. In the interest of time, I think I'll skip this. So then the next thing that we like to evaluate is the antigen-specific T cell response, and that's where the MANAFEST assay comes into play. So basically, this is a 10-day T cell culture with predicted neoantigenic peptides as identified by whole exome sequencing. And then at the end, we do TCR-seq on every single peptide stimulated T cell culture. And we have a core facility here at Johns Hopkins that runs this. So, you know, shameful self-promotion, if anyone is interested in doing TCR-seq, we do that. And we'd love to chat with you guys about that. And we really did that because, as you can imagine, this assay is quite pricey if you're doing your TCR-seq through, you know, standard commercial vendors. So we run all of this in-house, and we've really gotten this kind of streamlined over the last couple of years. So basically, the MANAFEST assay is the combination of the T cell culture with the TCR-seq, with then a bioinformatic pipeline to identify the individual clonotypes that are recognizing the antigen of interest. And we have used this assay in a variety of settings. We initially did this comparison between MANAFEST and conventional immune assays, and we were able to recapitulate results where ELISpot responses were high and robust, but we were also able to detect clonotypic expansion in cases where we could not detect ELISpot responses. And then just, you know, to give a shout out to ProImmune, we used their pentamers to validate this technique, and clonotypes that were pentamer positive were ones that were identified to be positive by our FEST assay in response to EBV antigen. So again, these are, you know, robust, high frequency responses that, you know, there's no mistaking. And I think 3 of these are actually public TCRs. So they were already identified to be, you know, positive for EBV. So just a real world example of the MANAFEST assay to detect neoantigen specific responses. We published this in New England. So again, this was from the Neoadjuvant study. We identified 3 TCRs that were specific for this neoantigen number 7. And this is a patient who had 0% residual tumor at the time of surgery. We then tracked these clones in the periphery, and we were able to track the clones in the pre-treatment and resection specimens. So just an example of the types of things we can do with the assay. When we perform this assay in a larger number of patients, I'm showing 6 patients here, but we've done this on, I think, 10 of the patients from the study so far. What we're showing is the average frequency of the positive clonotypes after T cell culture. So in blue are the patients with major pathologic response, and in red are the patients who did not respond. And basically, you know, this is a lot of dots on here, but basically what I'm showing is we're able to detect neoantigen specific responses regardless of the patient's response to PD-1 blockade, which is not something that has really ever been described in metastatic cancer, let alone in the resectable setting. So, you know, these are patients, they don't necessarily have high disease burden. They don't have oligometastatic disease. These are patients with relatively small localized tumors, and we're able to detect these responses using peripheral blood T cells. So here are just some examples of you know, this kind of thing. On the left is this patient again, this patient with 0% residual tumor. And what I'm basically showing is all the TCR clonotypes that expanded and the frequency of those clonotypes in all of the different wells. So the axis here is the putative neoantigens that were tested. Here's our famous neoantigen 7 that's been reported previously. And then here we have all of the TCR clonotypes that were identified by our platform. On the right, this is our patient who did not have major pathologic response. And as you can see, we actually found even more TCRs that were positive for neoantigens. And the major question is, you know, how do you know these are real? How do you know you're not just picking up, you know, high background or whatever? We clone almost all of these when we can, if we're able to get the alpha chain using single cell technologies. And as you can see, you know, these are validated bona fide neoantigen specific TCRs that we're looking at. So these are not false positives. So then the kind of holy grail is to link the neoantigen specific T cells with their functional programming in the tumor. And again, that's using the TCR as a molecular barcode. And I'm just kind of giving a little snippet here because, again, I could talk about this for 5 hours because of the amount of data that we've generated from these patients. But we have done single cell on, I want to say, over 300,000 cells from this patient cohort. And this is specifically T cells, so 300,000 T cells. What I'm showing here is a UMAP of only the CD8s because that's what we're identifying by MANAFEST. And using the MANAFEST assay, we are able to not only identify neoantigen-specific clones, but also clones that are recognizing viruses. And we know that viral-specific clones are prevalent in tumors for whatever reason, we still don't know. But we're able to compare the transcriptional programming of these virus-specific T cells with neoantigen-specific T cells, and you can see that they cluster very differently. So all of the neoantigen-specific T cells tend to reside in tissue-resident memory clusters. Our influenza-specific T cells, which we think of as the quintessential T cell response that resides within the lung, which is important for lung cancer. You know, these cluster separately into an IL-7R high tissue resident memory population. And then our EBV specific cells are also separate. So I think this is really interesting, and we're in the beginning stages right now of analyzing these data and understanding what it all means. But this is just an illustration of the types of things that this platform can do. And then the last thing, this is again just a little tidbit, is we're also able to look at CD4s and Tregs as well, and we have an entire effort to look at antigen-specific Tregs in these patients because we have noticed that in the tumor of our neoadjuvant patients, our treated and untreated patients, there is a higher frequency of Tregs in non-MPRs and untreated patients. When we look at EBI3, which is part of the immunosuppressive IL-35 heterodimer, we also see a significant increase in non-MPRs and untreated patients. And then when we look at GARP, which has been reported as being a marker of antigen-specific Tregs, we see the significant decrease in our MPRs and untreated patients. So what we think this is, you know, if you look at these UMAPs, EBI3 and GARP are mutually exclusive for the most part. We think we can have these non-redundant, mutually exclusive mechanisms of immunosuppression in the non-MPRs. And given the GARP expression, we think that there could be some element of antigen specificity with the Tregs. And how does the single cell all play into this is that once we identify T cells with GARP or EBI3 or with features of interest, we can clone out the TCRs because we'll have the alpha and beta from the single cell, and then we screen the TCRs for recognition against a library of class 2 antigens, and this is something that we have ongoing right now. And then of course we can also look at the dynamics of these T cells and their anatomical distribution in these patients. So the last thing that we hope to do is associate 1 and 2 with clinical outcome. And I always have this to be continued slide here because really we're in the relatively early stages of this, and we don't think that any of these things are necessarily going to be a biomarker that can be practically used in the clinic, but we think that it will identify new candidates for IO combinations and will identify you know, mechanisms of response or resistance to IO. So moving forward, our goals are to use the data that we have and leverage, you know, all of these really cool data sets and biospecimens to identify novel targets for IO combos. We want to develop a relational database that will allow us to really mine these data and make associations with clinical outcome and response to treatment. We, of course, want to define functional programs that are associated with response and resistance. And we really want to use these findings in the neoadjuvant setting, which is really attractive scientifically, to then see if our findings can be applied to the metastatic setting as well, and you know, and work in a larger number of patients. So with that, I really want to thank all of my collaborators, specifically Jiajia Zhang and Justina Kaushi, who did almost all of the work that I've shown here today. All of the folks in the Bloomberg Kimmel Institute, especially Patrick Ford, who always gets us all of the biospecimens from the patients. our genetics collaborators, and our bioinformatics folks as well, and of course the patients and their families for participating in research. Thank you.
Great, thank you so much, Kelly. That was a huge tour de force of single-cell-seq and a huge amount of data as well. So thank you so much. We have a question. I'm just going to ask Hilario to ask his question.
Great, thank you. Can you hear me?
We can.
That was an excellent talk, Kelly. I think lots of really interesting stuff going on there, really cool technology. I kind of got honed in on the end there. I'll talk a little bit about it in my talk around some of these antiviral T cells that you're finding with different functionalities by the TCR sequencing. So that's really neat and interesting. But I'm thinking kind of from a therapeutic approach, you know, how long does this take? And do you think there's any sort of potential here to help facilitate any sort of personalized medicine for patients here as you ID their T cells?
Yeah, so that's also another effort that we have ongoing that I didn't touch on today, but it's a very good point. We are using this assay to identify TCRs that could be used in what I would call off-the-shelf personalized T-cell based therapies, which I know is kind of like an oxymoron, but you know, the goal is to identify TCRs in patients at first with common HLA haplotypes that recognize recurrent mutant oncogene neoantigens. So these would be things, you know, like AKT1, P53, you know, all of the common oncogenes. We're hoping to generate a library or database of TCRs that recognize these for downstream T-cell based therapy. In terms of the, you know, general manifest single cell stuff, I don't really see an avenue for that clinically. I just think it's too expensive and too involved and too labor intensive, and it takes too long to ever be meaningful clinically for the patients that we're getting the biospecimens from. But where it is paying out is identifying new therapeutic targets and new candidates that will be used in the clinic hopefully very soon.
Great, yeah, thank you, Raj.
Thank you, Hilario, for your question. And again, thank you very much, Kelly, for your talk.